Hello, welcome to Learning Image Analysis. My name is Lawrence Small, and this is Lesson 4 of Section 1, Image Analysis Workflow. We'll be talking today about how to go about developing an algorithm. We'll be reviewing a lot of what the purpose of the different steps of an image analysis algorithm are, so that you can be prepared as you approach the process of developing that algorithm. So we'll start with talking about your measurements. What do you actually want to get out of your images? This is important always to do first, because it guides a lot of the constraints or freedom that you have when you go about analyzing the images. We'll then talk a lot about the stages of an image processing algorithm, the purpose of each step, and how they chain together so that you can have one in mind, the next one in mind as you're working on the earlier one. And then we'll touch on the process of algorithm development, how you go about putting one together, getting it to work on multiple images. Let's start with deciding your measurements. As I mentioned before, the measurements that you want to get out of your image, out of the features of your image, will define a lot of the flexibility and constraints that you have on your segmentation. So for instance, in this example, if we wanted to get the size of these particles, one problem is that the particles overlap. So it's hard to get the full area of the particle, or the circumference, or one of those sizes, directly from the segmentation. What you can do is get the diameter. So there's a measurement called ferret's diameter, or caliper diameter, which measures the longest length across the feature. And with that, it doesn't matter if some particles have overlapping portions. So it doesn't matter if this chunk is taken out. You can simply measure the longest diameter and get an accurate representation of this particle. So that's an example of something that is made much simpler once you know what your measurements are. Here's another example. Say we wanted to get an estimate count of the cells in this image. If all we care about is a count, we don't care about labeling each cell body perfectly. So these are pretty rough, globular segmentations here. But that doesn't matter. And if you know what your margin of error is, then perhaps you can take a little bit of imprecision. So maybe it's not that big a deal if these potentially 3 cells are all labeled as 1. If on the whole you've got an accurate estimate of the number of cells in this image. This is something that becomes pretty important as you start dealing with hundreds of thousands of images we've seen sometimes, where the exact number isn't so important. So, of course, knowing that ahead of time gives you a lot of flexibility and a lot of freedom in developing your image analysis algorithm. Another thing to consider is what constraints you might have. So there might be a particular standard that requires you to do point counting, or in this case a standard that requires measurements here along this adhesive layer at 25%, 50%, and 75% length. So in order to conform to that standard, even though we could get the exact thickness of this adhesive layer all across the image, well, we need to find some way to get the 50, 25, and 75% length. It really is pretty cool what you can achieve using some clever manipulations. So now let's talk about the stages of an image processing algorithm. We'll give a brief overview of each stage and what the purpose of that stage is. Later in the course, each one of these stages will be given its own full section. So we'll be going into a lot of detail later. Here I just want to give a brief overview of what's possible in each stage of the algorithm so that you can have that in mind when you're performing or when you're developing previous stages. So the first step is preprocessing. This is where you actually manipulate the grayscale values. You can do this to here remove noise. You can do this to highlight things that you want to find. So in this instance, if we're trying to identify these grains and measure their width, then we want to highlight the edges so that the boundaries of the grains can be found even though they've got different grayscale values within them. You couldn't just take a regular binary threshold. So you can use things like texture filters to find these desired features. You can use other filters to remove distracting features, just to give you something that you can then process properly. Once you have your preprocessed image, you can then produce a binary segmentation of that image. And by binary segmentation, I mean a version where some features, some pixels are selected and other pixels are not. So here we have the red pixels selected and the empty pixels unselected. There's many different ways that you can select certain pixels. You might use a basic threshold where pixels above a certain gray value are selected or below a certain gray value are selected to find these interiors without finding the boundaries. There's more advanced ones such as adaptive thresholding, such as watershed segmentations, and we'll talk about all those in the section 3 on initial segmentation. But just know that there's many different ways that you can take this and produce the initial segmentation. It's important to remember, though, that it's okay if this initial rough segmentation has errors, because we then have segmentation manipulation that you can perform afterwards to fix those errors. Things like removing noise, little things that were selected that shouldn't be, adjusting the size of the features to make them closer to the original grayscale value features that you want to find. You can even do some manipulations, replacing features with their major or minor axis, replacing them with perfect circles, replacing them with boxes, all in the process of the manipulation steps. And you'll see an example of me doing that kind of manipulation here in just a second, and the power that you can get out of those kinds of replacements. And lastly, we have binary mathematics, where you take information from two different rough segmentations and use them to inform each other. So finding, well, say this is a feature in one, so it shouldn't be a feature in another. And again, I'll show you an example of us doing that kind of logic here in a minute. So here's an illustration of a couple of the things that you can try to do when adjusting a segmentation, rejecting small features, joining line breaks, these kinds of corrections that can be made on the rough segmentation to bring it closer to the original features. And here's an example of that happening using what's known as a watershed algorithm to cut these lines apart. Here, 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 cut all these, and then using a cleanup step to shrink all these boundaries to 1 pixel wide. So now this represents this entire grain. And here's an illustration of some segmentation mathematics, binary mathematics. Say we have a rough segmentation that finds little particles, but it also finds this mess around the outside of large particles. Now, previously what we had done is we had found these large particles. We can take these large particles and, using mathematics, subtract them away from this rough selection of the small particles. And then all that's left is the small ones. So manipulations like this really help you isolate in on certain features that you want to find and remove things that aren't relevant to your image. Here's another example. Instead of doing a subtraction, we're actually looking for mutual features. So we have two selections. First, finding all the bright stuff. Bright stuff are silver ions here on the bottom of this adhesive layer. Then we have a second segmentation that finds this adhesive layer. And then I dilated it out, expanded it so that it overlapped with the particles a bit. And the result is, if you keep mutual features, you can reject all this nonsense down here at the bottom. And you just have the silver ions that are between the adhesive and the dentin here. And you can do the opposite, keep exclusive features, and just keep these things down here if for whatever reason you wanted to. Once you have your features properly segmented, then it's time to actually measure them. So there's many different ways that you can measure the segmentation. You can look at the entire image and measure, for instance, the area of the segmentation or the percent area, count the number of features. You can quantify the average grayscale intensity across the whole segmentation. You can also break that segmentation up into individual features. So for each grain within the segmentation, you can get its size, you can get its shape, you can get its brightness. So you had a bunch of cells, some were brighter than the others, you could classify each one. Those are per feature measurements. You can also take that segmentation and apply pixel by pixel measurements. So for a certain pixel, what's the thickness of the segmentation at that point? What's the orientation? Within a local window, what's the density of features present? And lastly, you can measure one segmentation relative to another. So you might have a region of interest where the cells are selected, and then you have another segmentation where individual particles within the cells are selected. And then you can count the number of particles within a cell. So that's another use for multiple segmentations that can be used to specify these measurements relative to one another. Now let's transition into MIPAR. And I can demonstrate the examples that I showed earlier and how the different stages of the image processing algorithm were applied to them. So if we start here with our beads recipe, remember that our desired measurement was the caliper diameter across each bead. So we start, of course, by removing these annotations. So that was just, they were found manually, and then we'll save that away for later. Now to the original image, we removed a lot of the noise with some pre-processing. So just a regular average blur. And then we segmented it using an adaptive threshold. Now we've got all of the segmentation manipulation where we remove small holes, where we do what's known as a morphological opening here, shrinking the features and bringing them back. And then we do separate features, a watershed, just like that, to separate those apart. And cut away anything that was next to our images here, or excuse me, next to our annotations. Right? The next thing we'll do is reject features that are here, have background here. So all the features that are really dark. So that's pretty simple, just rejecting features based on intensity mean. And then we reject features that are too small, so a bunch of nonsense here, and also features that likely don't have at least one full diameter present. Remove edge features because you don't want to measure partial features, of course. And then we're done. We have our feature measurements, and we can get the summary statistics like this, or we can actually view the histogram of measurements to see each feature and what its particular size was. So, just like that, and we see our mean caliper diameter of 1.26 micrometers. Let's move to our cells now. So we'll load up our cells image and the recipe to count them. And so, of course, our measurement is count. We don't care about size, we don't care about shape, we don't care about a lot of stuff that might put restrictions on how accurate the segmentation must be. So here, we'll of course do an average blur at the beginning to remove a lot of noise, and we use a different initial segmentation. We use an edge finding, so that here is going to find all the edges that are inside the cells. And that's something to think about. You want to try to think, what is different about the features that I want relative to everything else? And here they have a lot of texture, so an edge finding is able to do that pretty well. And then we use an erosion here to break all this up, remove all the tiny little features, and bring it back. And then we close up a lot of small noise. And lastly, we have what's known as a separate feature step again to cut these in two. Cut here, there was a cut here, a cut here, and a cut here. So again, pretty simple. Just clean the image, determine what you want to find, and use a segmentation step that finds that, and then get rid of all the nonsense that you don't care about, and do a final cleaning. And then you can count the number of cells that we have. Let's move on to our next image. So what we're looking at here is dentin, so this is a tooth, and here we have adhesive between the tooth and the compound used to fill a cavity. So here along the adhesive dentin boundary we have silver ions, and what these do is show where there's been gaps between the adhesive and the dentin, where that adhesion process didn't work properly. So these silver ions will infiltrate these gaps and highlight them. So if we wanted to, for instance, measure the adhesive length and say we have a standard that says we need to measure the width of this adhesive layer at 25%, 50%, and 75% thickness. So that's something that's pretty simple to do, actually. So now I'll show you how we do it. So the first step is to take this image and crop out the scale bar and all this information down here. Rather than just do a manual edit, figured for demonstration purposes, it could be a little bit fancy. So we first draw a single line along the bottom, and then we do a threshold here that highlights everything that's black, and then keep mutual in order to find this information here. Fill the holes and invert, so now we have everything else, and then we do an automatic crop, so that removes the scale bar automatically. Now to the pre-processing step. To help get these boundaries properly without having our segmentation sort of snake in where it shouldn't, we use a blur. So this is a low pass Fourier transform filter, and we'll talk about that in segment 2. But this is our pre-processing step, and then we can do a basic threshold to find the adhesive layer. Of course, now we have the segmentation manipulation, so we keep just the largest item here, which is the adhesive layer. A little bit of cleanup, and that's our adhesive layer. So now we get to the fun part. which is identifying in this layer what's the 50% and the 25 and 75% thicknesses, and this is all done with segmentation manipulations. So if I show here in outline mode, the first thing to do is replace it with a box, and replace the box with a major axis. So now we've got a straight line straight across the adhesive layer. Now we can replace this line with its centroid, and that's our single point. So note that this is the actual point. I had the outline too thick so that you could see it when zoomed out. So we take this centroid and subtract it from the line here, from the major axis. And now each of these we can replace with their centroids. So that gives us two of them, two little dots here that we can combine with the previous one. And now we have single points at 25%, 50%, and 75%. Now we can take our original box and subtract the points out and use the same separate features step that we used previously to cut this box into pieces. So now what we have are these three separate boxes. Then we've got some cleanup here, which I'll talk about later, but we subtract these boxes from the original one single box to get just these ribs that go across. Right? Now you can take these and take our original and do an intersection between them, and you get the widths at these points. So we take these ribs, intersect with the original, and there we are. And all this is automatic. So if we go back here to the manual edit step, and if we were to chop off a chunk of this, just like that, off of both sides, why not? Then the replacements and all the manipulations will readjust, and we have the same ribs that we had before. So the one thing you have to worry about is that sometimes when you do these separate features, you'll get cuts across, but that can be figured out as well. So what we'll do is take our points here. So we've got these horizontal and the vertical lines, and just reject them based on the orientation. That gets rid of all the horizontal ones. So lastly, we add the single points back in, which I didn't talk about before. And then we have our ribs and our intersections. And again, you can adjust these to be whatever you wanted, as long as the major axis was in the horizontal direction. So this particular recipe is limited. If we were to go this way, it wouldn't work quite right, because their first initial replaced with major axis, would go vertical instead of horizontal. So it's a limit to this particular recipe. Alright, now next what we'll do is identify just these silver ions in the cavity. So if you wanted to tell, well, how much infiltration was there, how bad was the adhesion between the adhesive layer and the dentin. So we start with our, again, cropping away the information at the bottom. And now we're going to use our smart cluster, also known as a k-means clustering, if you're familiar with that, to pre-process our image into 4 different classes: the dark stuff, the bright stuff, and all the stuff in the middle. So we have our adhesive layer here, which is the dark stuff. And again, now we're going to dilate this out a bit so that it overlaps the silver ions. And then we go back to our pre-processed image and find the bright stuff. All this bright stuff here. Okay, so that now we do a segmentation manipulation to only keep the stuff that overlaps with this. So only keep the mutual features, and that finds our silver ions. So it's simple as that. So now let's go back to our presentation. And we can talk about the process of algorithm development. So as I mentioned before, the first thing you want to do is decide what your measurements are. Once you've decided your measurements, then you can look at the image and decide what you want your selected features to be. Okay. Once you have a target in mind, then you can go about the process of pre-processing, initial segmentation, rough segmentation, segmentation manipulation, until you find the features that you want. Then you can go back and find perhaps a different class of features and continue that until everything that you want in the image has been found properly. Then the hard part is to load up another image that you have and adjust the recipe so that it's more robust, so it actually works on multiple images. Now there is a second way of doing it. Rather than doing the whole recipe at once on a single image, you can take each stage and do them on multiple images at a time. Either way can be effective. It's just really a matter of personal style. So a couple of lessons that I've learned through the process of doing lots and lots of image analysis algorithms is that development is going to be trial and error. You're going to have to use different ways of segmenting, different ways of cleaning, different pre-processing filters to highlight the different things you might want to identify. So don't be dismayed if it takes several times, several different methods to find something that actually works for you, and be willing to move on and try something else rather than just sitting and staring at the screen for half an hour. Also be aware that you don't need to be perfect, especially with your initial segmentation. You can clean it later, and you have some sort of error that's acceptable in your experiment. You only need enough accuracy in order to be able to tell your experimental group from your control group, for instance, or to be within the accuracy specified by your particular standard. If it's not perfect, that's okay. Having in mind what cleanup operations are available later will help you make decisions on what types of errors are acceptable in your rough segmentation. So knowing that you can reject small features or reject large features, and knowing that you can connect dots, knowing that you can separate dumbbell-shaped features into 2. These sorts of operations really give you a lot of freedom to initially process the image. And lastly, I will say the experience is the best teacher. My boss is much better than I am. I've seen him take images with rulers in them and accurately find 1 centimeter within the ruler, ignoring the holes in the middle of the ruler and the millimeter marks, so that he can have an accurate scale bar to normalize later measurements too. So you'll find eventually that almost anything can be found with enough practice and with enough trial and error. Next time, we'll start section 2, which is pre-processing, and dive deep into grayscale manipulations of your image. So some homework that we have are to collect images that you want to process. So we'll have a couple of example images available, but it would be really good if you had some of your own. And I recommend having several different types of images, so not all of the same thing. And of each type, have several examples. That way you can get used to making algorithms robust. And of course, have a clear idea of what you want to measure in your images. So when I say what you want to measure, I don't mean like a feature, like I want to measure the cells or the layers or the pores, but have a quantifiable characteristic. So I want the number of cells, the width of the layers, the area fraction of the pores. These kinds of things, that way you can have the most explicit direction for your actual algorithm development. And of course, don't pre-process your images. So if you're sending something that's been run through the sepia filter on Instagram, it's not going to be the best. So unless you specifically want to measure the effects of that adjustment. So it's not that we're not going to need these adjustments. That's what pre-processing is these grayscale adjustments, contrast adjustments, that kind of thing. But we want to start from scratch. And we'll discuss those filters in section 2 if they're really necessary on your particular image. So we'll see you next time.